if scientists just discovered that the part of your DNA they thought was useless garbage is actually the most dangerous thing on Earth, and they already started editing it? For decades, genetics operated under a convenient assumption. Nearly 70% of the human genome was labeled junk DNA, biological static left over from millions of years of evolution. But recent research has overturned this paradigm, revealing that this vast, unmapped territory acts as a master control system governing how and when genes across the entire body are expressed. Tucked inside this dark genome is a startling presence. Researchers at the Salk Institute report that approximately 8% of our genetic makeup consists of ancient, dormant viral sequences known as endogenous retroviruses. This means we are not just editing human biology, we are manipulating ancient viral material integrated into our very code. When tools like CRISPR are deployed to fix a single mutated gene, they do not operate in isolation. A late 2023 study published in Nature Biotechnology revealed that a single CRISPR edit can trigger unintended cuts and ripple effects across up to 1,500 seemingly unrelated sites. The intervention is not a clean, isolated surgical strike. Instead, it is a network-level disturbance. By introducing a single cut, we risk flipping biological switches and potentially reactivating dormant sequences that have slept for millions of years. Every confident edit we make today is an active gamble in a genomic system we are only beginning to decode. In March 2022, the Telomere-to-Telomere Consortium completed the first truly gapless human genome sequence, mapping the remaining 8% of our genetic code that had escaped scientists for decades. This newly charted territory added 200 million base pairs to the human reference map, resolving the highly repetitive stretches of centromeres and telomeres that direct cell division. Within these previously dark regions, researchers identified over 2,000 new gene candidates and at least 115 new protein coding sequences. Some of these newly mapped sequences resemble genes from organisms that have been extinct for more than 300 million years, preserved like genomic fossils inside living human cells. The completion of this map changed our view of human biology, turning what was once a blank space into a dense forest of active genetic material. But mapping these structures is not the same as understanding them. While scientists can now see every letter of this previously hidden code, they do not yet know what instructions these new genes carry, how they coordinate with the rest of the genome, or how they will react when modern gene editing tools are introduced. Visibility has made the physical terrain clear, but the functional rules of these newly exposed regions remain largely unexplained. We have acquired the complete map of our genome, but we have yet to translate the language in which it is written. In 2023, researchers at the Broad Institute conducting CRISPR experiments on human embryonic cells documented a concrete genomic warning sign. In roughly 14% of the edited samples, the gene editing process accidentally reactivated dormant retroviral sequences that had been silent for millions of years. The experiments demonstrated that targeting a single site could inadvertently release the epigenetic brakes on this ancient viral DNA. Once reactivated, these sequences did not remain passive. They initiated transcriptional programs that disrupted cell cycle regulation, causing the affected embryonic cells to behave in ways that closely resembled early-stage cancerous transformation. This transition from theoretical risk to documented cellular consequence triggered immediate concern within international bodies. In early 2024, the World Health Organization quietly convened an emergency advisory panel to evaluate these unintended genomic activation risks. While the panel's deliberations received virtually no mainstream media coverage, the scientific testimony warned that existing safety screens remain blind to these hidden genetic switches. Commenting on the profound gap between our ability to cut DNA and our capacity to predict the biological outcome, panel member Dr. Françoise Baylis warned that scientists are actively editing a book while still learning the alphabet. On December 8, 2023, the U.S. Food and Drug Administration approved Casgevy, a CRISPR-based therapy for sickle cell disease. This decision transitioned gene editing technology from a controlled laboratory environment into active clinical medicine. offering potential relief to thousands of patients. But the agency's review process focused primarily on validating the intended genetic cuts, rather than mapping the full, unpredictable cascade of genome-wide effects. The regulatory evaluation accepted localized success at the target site without requiring exhaustive, genome-wide surveillance of the newly mapped dark genome. This approach exposes a critical policy gap. A 2024 analysis published in the journal Science warned that current regulatory systems are structurally mismatched with the multi-layered nature of genetic interventions. Designed decades ago for simpler, single-gene therapies, these systems are unprepared to evaluate tools that can interact with thousands of genomic regions simultaneously. By treating gene editing like a traditional small molecule drug, these legacy guidelines risk ignoring the potential for long-term genomic instability. Despite these basic uncertainties, the global clinical pipeline is accelerating. At least 50 active CRISPR clinical trials are currently running worldwide, administering these therapies to human patients before the long-term safety science is fully established. This rapid expansion means that human subjects are receiving permanent genetic modifications, while the functional rules of the non-coding genome remain unexplained. The clinical race is moving forward, leaving public health regulators to govern a biological frontier using an obsolete playbook. In November 2018, the theoretical debate over modifying human DNA collapsed into reality. Chinese biophysicist He Jiankui announced the birth of twin girls, Lulu and Nana, whose embryos he had edited using CRISPR before implantation. This unauthorized clinical procedure bypassed global safety consensus and resulted in a 3-year prison sentence for He. Today, these children remain at the center of an unresolved biological debate, representing the first real-world case of heritable human genome editing. Despite his release, international scientific consensus remains firm. An international commission statement in 2023 held that no country currently has the technical ability to safely perform heritable human genome editing. The primary danger lies in the unpredictable. Unintended modifications within the dark genome may not manifest for 1, 2, or 3 generations. Because these edits alter the germline, any hidden errors are written permanently into the family lineage. Currently, the scientific community has no established roadmap for monitoring these multi-generational outcomes, leaving the biological trajectory of these children entirely unguided. Leading researchers are not calling for a complete halt to genetic medicine. Instead, the scientific community is advocating for a targeted moratorium specifically on heritable germline editing, while allowing non-heritable somatic therapies to proceed under strengthened regulatory oversight. The core demand is to prioritize a complete, functional map of the dark genome before any further heritable modifications are attempted on human embryos. This mapping effort is expected to take a decade or more of intensive laboratory study. Currently, national laws are highly fragmented. While the United Kingdom, Germany, and Canada have established domestic prohibitions on germline modification, they possess no legal authority to stop or police work conducted in other jurisdictions. Because genetic technology easily crosses borders, independent regulatory systems remain fundamentally limited. Policy bodies warn that the next 5 years represent a decisive, closing window to establish a binding, international governance framework. If nations fail to coordinate a unified response within this timeframe, heritable gene editing capabilities will permanently outrun the global ability to regulate them. With calls for an international moratorium growing, we face a sobering reality. We are rewriting the human book of life before we've even learned its alphabet. These permanent changes will echo across generations. Are we truly ready? Let me know your thoughts in the comments.